of today's lesson. So at the end of this lesson, we will be knowing the main characteristics of fungi. We will know the general study of moles. So we should know that is very informative. You should know about the basic study of moles. We should know the structure of fungi, nutrition, respiration, reproduction, and we are going to deal with the main part, that is economic importance of fungi and few harmful fungi. So please subscribe to my channel if you want to know all regarding these things which I am going to discuss in today's lesson. Subscribe to my channel, and if you are going to if you want to know more regarding the upcoming lessons which I am going to teach you, you need to stay tuned and stay updated with whatever videos I am teaching as per your curriculum. So don't forget to like and share my videos with your friends, and let us start with today's episode. Now, first let us do a quick recap of what we know about fungi. So what we know about fungi, we know that fungi, they do not contain chlorophyll, so they are non-green. They are plant-like multicellular organisms. So let us write that. So they are non-green and they are multicellular organisms, right? They are multicellular organisms and they are plant-like organisms. So these are the facts which we all know till now. So they generally grow in dark and moist places. This also we know, this is one of the main characteristics of fungi. We can say that they are growing in dark and moist places. Okay, so they are growing in dark and moist places, and actually the substratum containing dead organic matter, and that is they are also growing on dead organic matter. So they are growing on dead organic matter. So apart from this, we can say that on the bread, on the fruits, on leather, we can see some whitish thing that is coming up. That is also fungi, and that is also seen on the bark of trees. So we can see fungi mainly on the food, on bread, okay, on bark of trees. We can see that on bark of trees also, at times we see that moles are generally observed, and we can see them on, if you keep a leather bag, leather jacket or leather shoes for a long period of time in a dark and moist place, you will see some moles or whitish thing is coming on that thing. So that is the fungi, that is the mold. So on leather also it is present, it is found. So if we are keeping these things or storing them, preserving, not rather preserving, if we are keeping them for a long duration of time unattended, so on that only these molds will grow. Now let us see on the other features, characteristics of fungi. So first we can say that these are eukaryotic organisms with nuclei and mitochondria. Now they have a definite cell wall and made up of fungal cellulose. So what is the cell wall made up of? It is made up of fungal cellulose. So we should remember that we have seen that cell wall is also made up of cellulose. It is made up of chitin also, but here it is made up of fungal cellulose. Now these fungi, they do not contain chlorophyll. As they do not contain chlorophyll, they are non-green in nature, right? And as they are having no chlorophyll, they are heterotrophic. So if they were having chlorophyll in them, they would be able to produce their own food. So then they would have been autotrophic, but in this case they are heterotrophic. They are non-motile and they cannot move freely. So they are not moving freely from one place to another. They can reproduce mostly by the spore formation. So they are reproducing mainly by spore formation. So these all in details we are going to study. So these are the main characteristics. Apart from that, we have told that they are multicellular plant-like organisms. They are growing in dark and moist places and on dead organic matter. And they are also grown on food, bread, bark of trees and leather if they are being kept for a long period of time. So let us move on to the next part. Now here we will like to study a general information about moles. So what we are observing here, we can see that if the bread is kept for a long duration of time, or rather a few days after the expiry date is over, we will see that this common whitish thing will appear on the bread. So this is called bread mold. Okay, mucor and rhizopus, these both are most commonly found bread molds which grow on breads, and they are growing on other organic matters also, such as fruits also. You can see if it is kept on for a long duration of time, it gets rotten and it is having bread molds like fruits, vegetables when they are kept in warm and humid climates. So what is the ideal temperature for the growth of these molds? That is warm and humid places. And the main examples are mucor and rhizopus, which are the common bread mold. Now let us see. Now this is the structure of rhizopus. Okay, so bread molds are rhizopus and mucor. So this is the common structure of this rhizopus. So you can see that here this sporangium is there. This is sporangiospore. These dots are sporangiospore, and this is also a sporangiopore, and this are root-like structures. These are rhizoids. So this structure we will discuss in details. Now just keep in mind that they are growing on the fruits as well as on the bread. The cottony thing is the rhizopus or the mucor. Now, when we are coming to the structure of this mucor and rhizopus, what we are seeing here in this structure, we are seeing that bodies of mucor and rhizopus, they consist of tiny filaments called hyphae. So you can see that if we are looking at this structure, this diagram, just observe that these are tiny filaments which are called hyphae, and these tiny filaments, they contain cytoplasm and nuclei. You can see these blue dots, these are all nuclei, and they contain cytoplasm inside it. So cytoplasm and nuclei are present in this hyphae, and this hyphae we can say that the walls are made up of chitin here. These walls, they contain chitin, so the wall is made up of chitin, and the hyphae are the living growing part of the fungi, and a mass of entangled hyphae are known as mycelium. So this is the growing part of the hyphae. So this part is growing. So this is the live growing part of hyphae, and when this full mass is taking together, it is the entangled mass, and it is called the mycelium. Is it clear? So each growing part is called the hyphae. The cell wall is made up of chitin, and these tiny filaments, they consist of cytoplasm inside and with a nuclei. And these entangled hyphae, they are called mycelium. Is it clear to everyone? So they are called mycelium. So now we will move on to the next slide. Now in the last part, we have seen that a group of hyphae or a bunch of hyphae is called mycelium. So what this mycelium is again doing? This mycelium of rhizopus, it contains some finger-like hyphae, we have told, that penetrate into the substratum in which they grow. So those finger-like things, they are penetrating into the substratum, and these are called rhizoids. So these hyphae which are penetrating into the substratum, they are called rhizoids. And the prostate hyphae, this part of the hyphae over the substratum are called stolon. So these are the new things you are learning. So make it very much clear. So the part of hyphae which is entering the substratum, it is called the rhizoid, and the part that is the prostate hyphae over the substratum are called the stolon. So over the hyphae, it is called the stolon. So is it clear with you all? So that is rhizoid, and the part over the substratum is the stolon, and one hyphae which was having cytoplasm and nucleus in it, and they are the growing structures of the fungi, and when they are combined together in a bunch form, they form mycelium. Now we are here with the nutrition. So nutrition, what we can tell, that bread molds, they are heterotrophs, as they are obtaining their nutrition from the substratum on which they are growing. So we have all told earlier that fungi, they are having no chlorophyll in them, so they are depending on others for their nutrition. So they are heterotrophs, they cannot make their own food. So they are heterotrophs, and they are obtaining their nutrition from the substratum on which they grow. Now hyphae, they secrete certain enzymes on the food, and that break down the organic material, that is proteins, lipids and polysaccharides. Okay, so these are secreting enzymes on the food that are helping to break down the organic materials like proteins, lipids and polysaccharides. Is it clear? Now this food which is digested outside their bodies, what is that called? That is called extracellular digestion. So digestion which is happening outside the body. If the digestion is happening within the body, then we are going to tell it is intracellular digestion. Now moles then absorb this digested food through the cell walls of hyphae. Okay, the moles are then absorbing all the extracellular digested food through the cell walls of the hyphae. Is it clear? So heterotrophic nutrition is there, and all the food moles are absorbing this digested food through the cell walls of this hyphae. Okay, especially from which part of the hyphae it is being absorbed? It is the tip region. Okay, so the tip region of the hyphae, will help to absorb the nutrition. So heterotrophic nutrition, which is the digestion is happening outside the body, and the external or the tip part of the hyphae is helping the foods to the cell wall of the hyphae. Now we will move on to respiration. Now respiration in this moles, it is brought about by the presence of oxygen only. So this form of respiration, we can all tell that it is aerobic respiration. So aerobic respiration, what is aerobic respiration? The respiration which is done in presence of oxygen. So in presence of oxygen, the respiration is always aerobic. So what is the reproduction type? Now both sexual and asexual reproduction takes place in mucor and rhizopus. So asexual reproduction takes place by the formation of non-motile spores. So what are non-motile spores? These spores are not able to move from one space to another. So they are called non-motile spores, and this kind of reproduction is asexual reproduction. And what happens when sexual reproduction takes place? That is by the conjugation of similar gametes. So what is this conjugation of similar gametes called? It is called isogametes, same type of gametes when they are conjugating. So that is called the, we can say that is isogametes. Okay, so same type of gametes are combining together, that is conjugation of similar gametes or isogametes. Now here three stages of reproduction, three types of reproduction rather we can say in rhizopus. So in the life cycle of rhizopus we have seen that three processes. First is the vegetative, second one is the asexual, and third one is the sexual reproduction. It takes place. So vegetative reproduction, how it takes place? By the fragmentation method. So vegetative reproduction is taking place by the method of fragmentation. So it is how it is taking place. It is taking place. It develops separately, making the fragments of stolon, and which are making the complete mycelium by this fragmentation method. Now next comes the asexual reproduction, where the main thing is the formation of these sporangiospores. So what are formed? Sporangiospores are developed from the upper side of the rhizoidal node. So what is happening? The apical part is swelling up and it is giving rise to sporangium. And as a nuclei and cytoplasm, we can say that they move apically. So all are moving near the apex, so they are moving apically. Now the cytoplasm of the sporangium, what is it doing? It is differentiating into a denser peripheral region with we can say more of nuclei and the central columella region with fewer nuclei and more vacuoles. And you can see here, down here is the sporangium, from which are the wall of the sporangium has ruptured here after maturation, and the sporangiospore is coming out in the form of powdery mass. Now after getting a suitable condition and a substratum, each spore they germinate. So what they are forming? They are germinating into a new mycelium. So they are germinating into a new mycelium. Is it clear? So they are under favorable condition. They are taking the substratum and each they germinate into a new mycelium. Now after this stage, in this asexual reproduction process only, there is a stage where there is formation of chlamydospores. Chlamydospores. So what are these chlamydospores? These chlamydospores are nothing. They are formed under unfavorable conditions. So they do not develop under favorable condition. So they are actually an intercalary segment of mycelium which are developing due to the formation of accumulation of protoplasm and septate. Okay, so they remain dormant until the favorable conditions are returning, and they will only germinate into a new mycelium when there is a favorable condition. Now we are coming to another part, that is sexual reproduction. So what is happening here? We can see that two compatible hyphae, these two compatible hyphae, they are coming together. So in both the mycelia, small outgrowth has developed and they are forming progametangia. So what they are forming? Progametangia. So when they are coming in contact, they are forming this progametangia, and this gametangia they conjugate to form a multinucleated structure. So what they are forming? They are forming a multinucleated structure, that is a structure which is having many nucleus. So this many nucleus having this structure is called the zygospore. This, please follow my pointer where I am pointing you, that they are forming first the zygote, and this zygote is a multinucleated structure that is forming a zygospore. So how this is being formed? So plasmogamy is followed by karyogamy. Is it clear? So plasmogamy is followed by karyogamy process, which is in turn giving a structure to and zygote is formed, and from that zygospore is formed, and rest of the unpaired nuclei, which is not at all paired, it is degenerating. So it is degenerating and breaking down. So this is how the zygospores, they are enlarging, they are becoming thick-walled, they are becoming resistant to the adverse environmental conditions, and only getting on favorable conditions. They will only germinate into a new, we can say they are germinating into a new fungi, or they are germinating into a new mycelia. So the inner wall of the zygospore, it is developing into promycelium, forming the germ sporangiospore with germ sporangium, and which forms apically. So we can see here the sporangium is forming apically, and from this sporangium what is bursting out, from that we are getting the sporangiospore. Is it clear? It's a bit difficult and complex thing, so you just need to go through it once and just remember the terms and things what is happening in the sexual, asexual and vegetative reproduction. Now here we are coming with the main topic of this lesson is economic importance of fungi. So fungi, they are actually principal decomposers that reduce the polysaccharides into proteins in dead bodies of plants and animals. So that is the main function of fungi. They are reducing the polysaccharides and proteins in dead bodies into simple organic compounds. So how they are working in industry and all, we need to know that. So they are actually helping in enriching the soil. They are helping in the biochemical cycling of material. So what they are doing, they are helping in the biochemical cycling of material. So let us see quickly how they are helping us. So first we can take an example of yeast, such as Saccharomyces is important nutritional supplement as it contains vitamins, minerals and other nutrients. Okay, so we can say that it is very important yeast which is taken as a nutritional supplement, and it is containing vitamins, minerals and other nutrients. And mushrooms, they are very important and they are very rich in vitamins and proteins. So many of us, we are taking mushrooms in our food. We are telling that they are good sources of protein. They are also rich in vitamins also. But we should be aware of 2 or 3 types of mushroom like this. This is the fly agaric or Amanita muscaria. So this is very poisonous. And this death cap, the second one, though it looks like the normal mushroom only, it is very poisonous, deadly. It should not be eaten at all. So these 2 mushrooms which are highly poisonous should not be eaten at all. That is fly agaric and death cap. So which when eaten, it can cause death also. Now next comes the use of yeast in this wine industry. How they are used in wine industry, the sugar is actually, it is fermented by yeast to produce ethyl alcohol and carbon dioxide. So carbon dioxide here, it is given as a byproduct. So sugar is fermented by yeast to produce ethyl alcohol and carbon dioxide. So carbon dioxide is not the product, it is the byproduct which is solidified in the form of dry ice. And the wine is also prepared from, we can say it is prepared from grapes, while the beer is prepared from barley. So the yeast used for this fermentation process is Saccharomyces cerevisiae. So this is the yeast which is used in the yeast fermentation process. This is the Saccharomyces cerevisiae. Okay, so this is the yeast what is used in this process. Now it is only continued. So in the process of cheese production also, we know that yeast is playing a major important role. So penicillium and aspergillus are used for flavoring the cheese. These 2 are used for flavoring the cheese in cheese production industry. Now curdling of milk is also done by lactobacillus. The curd produced is separated from the whey, and the curd processing, we can say in curd processing, it is the moisture is removed from the curd, leading to cottage cheese. So in the cottage cheese production also we are using these fungi. So it is a beneficial part of fungi. It is the useful role of fungi. Okay. So salting, what is done then from the cheese? Salting is used to further remove the moisture, and it is preventing the growth of certain undesirable microorganisms. Is it clear? Now what is ripening of the curd? Ripening of the curd, it is actually keeping the curd at an optimum temperature and moisture is the ripening of the curd, so that we can tell that it doesn't get destroyed. Now in bread making industry, yeast is also added to produce the sponginess of the bread. So if you are not adding adequate amount of yeast, we will not get a spongy bread. So sponginess of the bread is actually due to the presence of yeast in bread. Now fungi, they are also used to produce several chemical compounds that are important for food processing industry, such as citric acid and gluconic acid. And citric acid is actually used to make soft drinks and candies also. Now fungi is having many useful role in medicines also, like molds are used as antibiotics to check the effects of disease producing bacteria. Penicillin is used to cure pneumonia, and we can say that it is produced from molds. So many enzymes such as pectinase, amylase and oxidases are synthesized by fungi, Penicillium and Aspergillus. So these pectinase, amylase and oxidases are synthesized by the fungi, Penicillium and Aspergillus, and they are having the medicinal property also. Now here we are having some harmful effects of fungi. Now let us now discuss about some harmful effects. So fungi, they can attack the tissues of plants and animals and cause diseases. Now generally it is very common, we can see that some allergies, people are having allergies like sneezing, sniffing and respiratory problems when they are not actually they are not cleaning their bed sheets, their bed covers and all. So that bed sheets or bed covers or the places or the dresses which they are wearing has not been cleaned for a long period of time, and they are having mold spores in them, which is causing sneezing, sniffing and respiratory problems. Now it can also infect and poison the human diseases like athlete's foot, that is fluid filled blisters are happening and itching is there on the skin. Now ringworm, we all know that ring shaped skin lesions, and we can see that ringworms are there, that is also a fungal infection. Vaginal yeast infection is also, it causes burning, itching in the vaginal area, and these are also caused by fungi. Now many fungi, they are attacking the food grains and food products and they are spoiling them. Next comes the late blight disease in potatoes, rust in wheat, smut in corn and downy mildew in grapes. So these are all some common fungal diseases in plants, and we have till now we have discussed all the harmful effects as well as the useful effects of fungi. We have discussed in details about the reproduction, nutrition and the structure of the fungi. So this is here where I am ending this lesson. So stay tuned, stay updated with all my lessons. Don't forget to subscribe to my channel, and next I'll be coming with the nutrition lesson. If you want to know more regarding as per your curriculum, and you want to get ideas and views on the nutrition lesson, how I'm teaching, please stay tuned, stay subscribed to my channel, and please don't forget to like and share my videos. Thank you.